4% of the faculty members are overseas. And then we have a students population between 35 to 40,000, depending how many we recruit in a particular year. And then 7 to 10,000 of our students are international. Can you hear me clearly? And I have a habit of moving a lot. I'm trying to. So, BIFAR, the Birmingham Institute of Forest Research. It was established in 2014, and the establishment came to build a 3-year carbon dioxide enrichment facility. 3-year means outdoor in the field, rather than bringing trees inside the laboratory to expose them to higher levels of carbon dioxide to mimic the future, because our carbon dioxide concentrations are increasing in the atmosphere. So we wanted to predict the response of the forest into the future CO2 enriched atmospheres. Even though it's a new institute in the university, but it has expanded quite a lot. It expanded quite a lot. And now we work in 4 key areas, which is highly multidisciplinary, and that's the core ethos of the institute itself, to do international collaborations, multidisciplinary, cross-disciplinary research. Our research is within these 4 core areas, and the implications have local, regional, and global implications. Key areas are, we work in forestry in the global sector, so looking at forest functions under global change, whether it's drought or extreme flood events. And we have a specialist group which looks at the health of the trees in terms of invasive pests and diseases. We have an interdisciplinary research program that integrates both natural sciences and social sciences. And the key flagship experiment, which is the pest facility, which involves the manipulation of our forest with elevated atmospheric CO2. So I will be giving a lot of details in what we found so far, but I will briefly introduce these 3 areas. In terms of global research, we have many projects globally, but one of our colleagues, Adriani, as an associate professor, she is leading the monitoring of forest plots, permanent forest plots across the global south. And it's a very extensive program looking at carbon budgets in response to global change. And Adriani is entrusted to identify collaborations in Pakistan, whether she is able to establish permanent monitoring plots, maybe in the Himalayas nearby, if data could be generated so that it will become part of this network. And this is something we will discuss later, whether there is scope for Adriani to put her in touch with someone here. One of our colleagues, Professor Vinscouchi, who works on trees not only for carbon, but he looks at greenhouse gas emissions and how trees are affecting the exchanges of greenhouse gas emissions, particularly methane, because not only carbon dioxide, but methane and nitrous oxide are important greenhouse gases. So he has been working in Amazonia in South America, and now moved to Ghana and Africa. And recently he published a paper in Nature looking at how trees are taking up methane. So our soils and rice paddies and wetlands are producing methane that goes up into the air. But then the tree canopy itself, the microbes and the tree barks are taking up methane and consuming it. It's a huge service by the trees. And this is the first time that a study of this nature and this scale published just last month. So something to consider in terms of tree counts and how Pakistani forests might be contributing to this global methane sink. We have many projects in terms of international research in the forestry sector. I give you just one more example, which is a cost action of the EU. So that's a network project where researchers are allowed and facilitated to develop networks for larger projects, collaborations, and so on. The one that I'm mainly associated with, and I lead the UK side of this project, is called the Joint Effects of Climate Extremes in Atmospheric Deposition on European forests. And atmospheric depositions could be nitrogen oxides, reduced nitrogens, or sulfur oxides, depending where you are. So atmospheric pollution component comes into play. The project itself is to really standardize monitoring, to link manipulative experiments like the one we have, the phase facility, and there are drought and moisture manipulation experiments across Europe. So, and there are many more. So, another goal is really to link them up to rise to the challenge of taking climate change. And then to develop harmonized data sets of tree and forest ecosystems. And data science and image analysis is something that could quite rightly fit in here. Even though it's a European network, but anyone anywhere in the world could join it. But the link is here, and I can share the link as well. And there are 4 work packages. And depending on your interest, you could join a particular work package, engage in some of the online activities, but there is also calls for funding under the short-term scientific missions program that allows a month or 2 months secondment to any institution working there, and it's fully funded. We just had a deadline of our second round of funding calls, which ended by the end of August. And not many applications came through, so there is quite a bit of potential just to go somewhere and be there for a month or so and with some relevant experience. So it might be a literature review that you might undertake, or it might be a short experiment, some data synthesis work, because a month is not long enough for experimental work. So good potential, and you have to show your link of how you are related to forestry. So if you meet a result and make a case, then the management committee decides in terms of remunerations. In terms of tree health in our emerging research, there's a lot of projects. So we have a school of biosciences. I'm in the school of geography, and then we have many colleagues which work on tree diseases and pests and pathogens and how these trees might be responding to the change in climate and in terms of the incidences of pathogenesis. We have key projects. This one is quite unique, which is called MENDRA, which particularly looking at biotic and abiotic stresses in terms of pathogenesis and how the trees might be transmitting the disease informations through their progenies via seed productions, and whether the future forest might be more resilient and then have an immune system to, similar to humans to trigger, to protect themselves from emerging diseases. Something which is quite innovative, led by our colleague Estrella, and then Rob Jackson, who works mostly on bacteria, and Graham and another of my colleagues, John is the last name, who works on fungal diseases. Their expertise are not only related to forestry but agriculture as well. So they do work on tomatoes and GM plants and so on. We do encourage in our institute integrations of human sciences into natural sciences, because we can't do our job unless we engage the people who really handle these ecosystems. Whether it's a policymaker or whether it's an artist here like some of our key projects, one is, which is NUST, is our national funding body, National Science Foundation here. So it's prestigious projects to have. So our colleagues in human geography are looking at the ways of young people and how they reimagine landscapes. In Manchester, they went to immigrants communities and the young children who immigrated, asylum seekers, because they immigrated from different regions and they may have a view of forestry and treescapes and how they reimagine those treescapes when they see similar trees in England. So trying to reconnect young people with their natural environment. So quite an innovative project. We do work with the English department in Birmingham for Professor John Holmes, currently working on forest ecology and ventrification, for example. Another colleague, Sami, who is working with us, and she is creating musical compositions using the voices of the forest. When the wind blows through the forestry, you can see the voices that she makes those musical compositions. And then Claire, who is based at the University of Islamabad, and she has been working with us for the last 10 years now, using analog cameras. So some of the images are on her website, and using the analog cameras, I showed you those cameras in the little boxes to take images of the forest evolution through the growing seasons, from leaf flush all the way to the autumn colors. And during the COVID lockdown, when there were senior people living alone, and they really needed to engage with someone, so Claire was the one who connected those people through those analog images and connecting those lonely senior citizens to re-engage with the environment because they were not allowed to go out. And so expanded quite a bit, and we got quite a high community support in the region because this project particularly enabled them to engage and not get depressed, because there is a mental health relationship between humans and green spaces. In forestry, we define forestry as where there are a large chunk of trees. But when Claire started working with us, then her definition of forest is everything in a forest is a forest. And this is something we adapted in Birmingham, the definition. And that's the benefit of working with human sciences and artists to understand our nature and reconnect. And this is something that fits very well with biodiversity initiatives. So that was all a general overview of the institute itself, but if you need more information or if you could see the potentials for collaboration, so you get in touch. Now I'm going to go to my area of research and Biogeochemistry. So this figure shows the global sources and sinks of atmospheric CO2 from anthropogenic sources, mainly from fossil land use change, and of all that carbon dioxide that is emitted into the atmosphere. About 50% ends up building the atmospheric concentration. That's why our carbon dioxide concentration went up from 280 parts per million ppm to the contemporary levels of about 420 ppm that we experience today globally. One third of that CO2 which goes up from these anthropogenic sources is captured back by the land ecosystems, and in the land 62% of that sink lies in the global forest. So it's a free gift from nature to allow and ameliorate these building concentrations of the atmospheric CO2. And then about 25% goes and taken up by the ocean. So it's a huge sink, but that sink is not consistent across the different land ecosystems and tree types and plant types. This sink is currently predicted by the Intergovernmental Panel of Climate Change, IPCC, increase with increasing atmospheric CO2. The reason is photosynthesis, or high school biology, which is the process where plants capture carbon dioxide, combine it with water to produce sugars and carbohydrates and starch in the biomass that goes along the food chain all the way to humans and animals and everything else. So there are predictions that CO2 is a key regulator of photosynthesis. And if we increase the carbon dioxide concentrations, then the land plants should be photosynthesizing more and capturing more carbon. But carbon dioxide is not only regulated by the availability of CO2 alone. There are water, disease, and nutrients. And you will have seen many farmers here in the UK, they're fertilizing the land with nitrogen, phosphorus, and potassium. The reason is to really prime up photosynthesis. So in the IPCC in the 2013 report looked at the land carbon sink on the negative side in response to rising atmospheric CO2 concentration. This is where they predicted with the rising CO2 we will have higher CO2 uptake through photosynthesis and carbon sequestration. Individual dots represents the outcomes of individual models, the Earth system models that IPCC uses. And then the bar represents the mean of their carbon uptake. However, this prediction by the IPCC was challenged, and when the modelers looked at the availability of nitrogen alone, say for example northern temperate forest, here even in Pakistan if you go to northern areas, these forests are severely limited by the availability of nitrogen. So when nitrogen was included in the land carbon sink, we predicted carbon sequestrations and the rising atmospheric CO2 was reduced by about 25 to 50%. So how we do it now? It's a huge sink, but we don't know the true magnitude of this sink. And to do an experiment at scale that includes land ecosystems to be exposed to carbon dioxide is a very, very expensive exercise. But we need to do something, because even if we just consider the northern temperate forests in this region, that's about 3 billion tons of carbon dioxide sequestered annually in the region. The amount of carbon dioxide in northern forests which is sequestered is roughly equal to the CO2 emissions from human sources from the EU 27 nations, including the United Kingdom, and it's the amount that is emitted into the air due to global deforestation. So it's a huge thing, but it's highly uncertain. And with this question, no one did an experiment of this scale to see how forests in the temperate regions particularly would be responding to higher atmospheric CO2 concentrations. So this is the first of its experiment, which we established in 2017 in Birmingham. It's one of the 3 largest climate change experiments. There is one in Australia for eucalyptus forests, and the UK funding for Brazil. Now we are working with the Brazilian colleagues building another facility in Amazonia. It's not in operation, but in 2025 the Amazon phase will be up and running, because different forests will have different limitations and potentials for capturing carbon. Ours is for mature forests. There used to be pest facilities in North America, but those were mostly young plantations and planted on full agricultural land. Our forest is 180 years old. It was planted by the Royal Navy to produce wood in 150 years so that they could make ships, warrior ships. So this was the original intent of this particular woodland in the region. Here we established these space arrays, which is 30 meters in diameter. And we have 6 of those. 3 with elevated atmospheric CO2 concentration. So from the ground surface all the way to the top of the canopy, from the morning to the evening, we raise carbon dioxide by 150 ppm above the ambient concentration. This is the concentration which would be the norm by 2050 and beyond. Something we wanted to not to be far distant, so we could have gone to the end of the 21st century and could have raised it to 800 ppm. But we wanted to be a little closer so that the policymakers could see the implications in their own lifetimes, and they could see it's coming, so they could take actions in response to whatever policy recommendations we might come up with. And when I say forests of the future, so at the moment our forests represent the atmospheres of the 2050s. This CO2 movement. We have 3 phase springs in 3 controls with the same infrastructure. So one is fumigating carbon dioxide, the other is just fumigating air. And we fumigate about 20 tons of CO2 every day. The cost is gone up, so it used to be 100 pounds a ton before the gas crisis in the Ukraine war. Now we pay about 400 pounds a ton of CO2. It's about 2.5 million of our annual funding goes into the CO2 fumigations, and the wind takes it out. And then we add more and more. So it's a huge cost. And then we have 3 non-infrastructure control plots where there is no manipulation, so we could see how the natural system is responding without this manipulation of infrastructure. The original project was for 10 years, and it was supposed to end by 2026, but early this year we got additional funding, so we will go into 2030s. So it's the first experiment in the world to go into a second decade of fumigation, and this is important, and I will come back to that point when I show some data.
Why do you get people of organization, the one that is not involved by the
So we need technologies, and this is something I propose to the UAE government for their mangroves, because they don't have trees anywhere in Dubai or Abu Dhabi except the mangroves on the coastline. So we were proposing if they are willing to invest, we could have technologies to capture atmospheric CO2, purify it, and fumigate it. We are still considering our proposal. So yeah, there are technological innovations, we could do that. Our carbon dioxide used to come from fossil sources, from ammonia fertilizer plants. But with the gas prices in Europe, the prices went up to 600 pounds a ton. And now we use waste. So the food waste in England are collected by a large industry which digest the food waste through anaerobic processes to produce biogas. And in the biogas there is carbon dioxide. So they invested in the machinery to scavenge all the carbon dioxide, and now we're using this food source, food waste, CO2 to do our fumigations, and then purify it to high standards. So there are examples of these innovations. And there might be.
Are there any adverse effects on the environment near this forest due to the elevated level of CO2?
No, it's just probably priming up, so probably supporting, but by the time it's out of the canopy, it's highly diluted. And we see about 600 ppm, 550 ppm sometime indoors. So nothing to that effect so far. It's a small woodland, and the engineers did a smart job. So we have these 3 elevated CO2 plots with the colors, and the blue ones are the control one. But the wind direction is such that we don't get contaminations of our control plots. So here is the CO2, which during the brewing season you could see it goes to 550 ppm from 2017 all the way to 2023. And 95% of the time we achieve that target. And there is a whole set of CO2 sensors which enable us to maintain this embedded concentration. Sometimes you see these high sparks. This is probably high wind blowing and just delivering a high concentrated CO2 to a sensor or something. But that's what we achieved so far. So we have been very successful in a small test to separate these 3 patches from these 3. Not only CO2 fumigations, but there is a lot of researchers, more than 100, engaged in research in this facility. So you name it, every tree is tagged, and there are sensors like the carbon dioxide sensors, the moisture sensors, automated analyzers, leaf level collectors, and dendrometers. So we have students, so Anna Gardner was one of the PhD students who is a research fellow now with the institute. She used to climb up to the trees, spend 6 or 7 hours in this makeshift office with these analyzers to look at the leaf level CO2 exchange. So how much CO2 the leaf is taking when the CO2 concentration goes up. And then there are people on the ground, like one of my PhD students, Alex, who is using automated LIFO systems to measure the emission of greenhouse gases, carbon dioxide, methane, and nitrous oxide from the ground surface back into the atmosphere. So we could do a complete budget, what comes in and what goes out. And here are the results. So this is Anna Gardner's initial PhD work. And she looked at initial responses. So on average, she saw a 23% increase in CO2 uptake at the leaf canopy level. We increased the concentrations by about 38%, and the ecosystem response was up to 23%, and the maximum was 31%. So yes, these old trees, mature trees, 180 years old trees were able to capitalize and capture more of this carbon. Trees could capture carbon, but they may not store it, so it might go back into the air and they could do respirations. If they are struggling, they will invest this carbon in resilience, resistance, diseases, and everything, And when they do that, that carbon goes back into the air as CO2, like a tree respiration. So here we see a 23% increase, and we continue those measurements. By 2023, there is still that significant response of more CO2 uptake over time. So by the 7th year of the CO2 enrichment, the forests are still taking up, because we want to look at the long-term trajectory of these responses, because short-term responses might not be indicative of the long-term stability and resilience of the system. So it continues to take more carbon where it goes. So we have automatic dendrometers, which are covering the tree diameters, and every hour it measures the diameter, and it's wireless enabled, so we download that data, and we look at the expansions of the tree over time. The trees shrink in the morning because they pump water into the canopy, and by the evening it expands. So this diurnal shrinking and expansions, because hundreds of liters are pumped by individual tree into the air through transpiration. So we could account for the diurnal responses and then tease out the growth responses. And there was a HEC exchange faculty member from Bard University in South Kashmir who worked with us in this data. So this is one of the examples that there are opportunities. And he just became a co-author on this paper, which we published last month in Nature Climate Change. The figure from this paper shows the growth responses of the trees. 2016 was our pre-treatment year. In 2017, you could see that there is an increasing trend of dry wood biomass in these mature trees, and there is a year-to-year variation. In 2018 and 2019, we had moth infestations. These are little insects which were denuding, which were eating the leaves, so the response went down. If we were doing the experiments for 3 or 4 years, we would have concluded that the growth response subsided and there is no growth response. But after moth infestations, it went up and the growth response has increased. In 2022, we had a heat wave and drought, and there was severe soil moisture deficit, and the growth responses declined and it went up again in 2023. No individual years are statistically significant, but if we pool the data, then it becomes significant at that level. So long-term studies are really critical to capture the long-term dynamics of ecosystems. So we see this growth response not only in the trees, but we see it in the leaves, fine roots, reproductive bodies, and the exudation of carbon. It's a cocktail of soluble carbon exuded by the roots that I will show you later. And when we compare the control, the CO2 to elevated CO2 just for 2021, so that's a 10% increase and 11% increase in 2022. And that's about 1.7 tons of dry matter per hectare per year is more carbon taken up by these ecosystems due to the rising CO2 levels.
And these CO2 elevated CO2, what is the difference between them?
So here we add 150 ppm above the ambient. So if we are having 400 ppm outdoor, then we raise the CO2 levels to reach to 550 ppm, which would be the norm by 2050. So and we are going in that direction. So that's the difference. 150 ppm concentration difference made this growth responses. So there is more wood and more carbon captured in biomass. How likely to take the biomass is a question. If we keep it for long term, we will be mitigating climate change.
Definitely not every day the same amount of carbon will be used, right? You can get carbon over here. So it could be different. So will you increase 150 ppm every single time, or will you just put it to
Every time, so we have 24 sensors in each experimental array, which measure the CO2 concentrations from the bottom to the top in real time. And based on that ambient levels, whether it's a windy day or it's a stagnant and non-windy day, then we have an automated system which triggers to add another 150 ppm. When it reaches that threshold, it stops. So it's on and off. If it's a windy day, the system is constantly blowing CO2. If it's a quiet day, it's on and off from time, just to maintain that high levels. The growth responses that we have covered in our forest is about 16 grams per meter square per year, which is similar the growth responses of an experimental forest of Oxford University. You could see the growth responses of Amazonian forest. and eucalyptus space in Australia. So we are in the broader range of what was expected. So no mistake there, roughly. Now the tree growth, the litter per amount, the amount of litter that falls down in the autumn also increased by about 10%. It's not the leaf area index which is calculated based on measurements and remote sensing technology. So the leaf area index did not change, but the leaf mass area, because the leaf thickness increased in response to CO2, and that's mostly the leaf thickness that resulted in this 10% increase in litter power. So that's an additional carbon going into the ground, and we measure it by 3 times in reproductive bodies and everything on a regular basis. Interestingly, it brings me to the point about nitrogen. When the leaves fall just before the senescence, we call it, when the leaf falls just before that, the trees reallocate the nitrogen in the leaf to the nearby branches and twigs so that they could use it next year rather than losing the nitrogen in the litter crop. And you could see it here that every year when there is a litter crop, the amount of nitrogen in the litter is relatively lower than the amount of nitrogen in the control plots. So first evidence that the trees are trying to conserve nitrogen much more substantially because they could see the potential. This conserved nitrogen is reallocated to the new leaves the following spring to be photosynthesizing, because nitrogen is a key compound of an enzyme in the leaves that enables the photosynthesis to happen.
Means that dry leaves just fall off from the tree. They are not able to cross any photosynthesis.
No, because they are conserving the nitrogen at the moment. And because this forest source the nitrogen from the soil, so the trees are using different mechanisms to look for more nitrogen. And this is what we are testing, whether it would be the nitrogen that might be limiting the CO2 fertilization. So here we install root boxes in the ground. When we remove these insulating surfaces, we could see a root window, and then we could remove the perspex cover here, and we could access roots, we could measure different things. One of the things we measure is the exudation of carbon, this particle, this solution like a dewdrop. It's like a slushy that is exuded by the roots into the rhizosphere to enable the microbes nearby to eat it and decompose the soil organic matter to release nutrients so that the roots could take it. So it's a symbiotic relationship between microbes and the roots. We also use cameras and images from mini rhizotrons to take continuous images of root growth. So the one you might have seen, the mini rhizotron or images. So we could track a particular root, and based on those tracking using AI, we could measure the root turnover, growth responses, and proliferation of the soils. Another area for image analysis extraction. So we collected root exudates while the roots were still attached to the trees, and we got them to syringes filled with glass beads in a solution that is mimicking the soil chemistry. And then after 48 hours, we collected the exudates in here, a very smart bright research with the majority of this work, which contributed to some of the key papers. Then she would click the root that was used for exudation measurements, and we have a root scanner which scans the root so we could look at the root area, specific root length in many parameters using that image. And there is a particular software called WinRhizo which one uses to derive all the root parameters from those images. So that's another area that is relevant to image analysis. Overall, we see an increase in the exudations of carbon. Over the year, that's on average is 40% under elevated CO2 compared to control ones. What the tree takes, part of that carbon is exuded into the rhizosphere, and they don't do this for free. The trees are doing this particularly to prime microbes to give them nutrients back. When the carbon exudation increased, the nitrogen exudation did not change, which resulted in the carbon to nitrogen ratios to change from 13 to 17. That means in the C to N ratios of 17, microbes will be up and running after the soil organic matter to decompose to